OneMedRadio welcomes George Yu, President and CEO of Cyto Technologies. Cyto is developing the product IV3, a first-in-class medical device that allows early detection, capture, and targeted destruction of metastatic circulating cancer cells in vivo. Cyto will present at the upcoming OneMed Forum taking place June 26th and 27th in New York City. So thank you for joining us, George.
Pleasure to be here.
I want to start with a snapshot of the company. What makes the IV3 device unique?
Cyto was founded about 6 months ago, and what we've done so far is licensing a technology from the University of Arkansas that's essential laser technology that allows the early non-invasive detection of metastatic cancer cells in vivo. So what's unique about the technology is that we're using a laser that points to a blood vessel in the hand or in the arm, and then as cancer cells pass by in the bloodstream, the laser gets absorbed by cancer cells, and the cancer cells then emit a photoacoustic wave that we can detect through an ultrasound transducer, and we can literally count the individual cancer cells that are clearly separated from the background noise that are typically red blood cells.
So I understand that the only option that's available right now is in vitro detection, so why does that come up short?
That's correct. So the current technology that's existing on the market are all in vitro assays or technologies. And the issues with in vitro detection is that you can only take a small blood sample, let's say a milliliter of blood, and you limit it in terms of sensitivity by the small blood volume. So the human blood volume is about 5 liters, and you take 1 milliliter of blood as a sample for in vitro detection, and you only detect one cancer cell in 1 milliliter. You cannot detect higher sensitivity than that. That 1 milliliter actually translates into 5,000 cancer cells in the body if you have 5 liters of blood volume in the patient. So you can really not detect less than 5,000 cancer cells in the body. And at that point, it's typically too late. You already have metastatic cancer manifested itself in the body, and you can't really intervene. And a lot of physicians would like to be able to detect metastatic cancer cells a lot earlier at a much lower concentration of blood volume. And that's why we use a technology that approaches a in vivo technology, where you can literally measure the entire 5 liters of blood volume. So if you use our laser technology pointed to a blood vessel in the human body, and you wait about an hour, and you measure the entire 5 liters. If you detect 50 cancer cells within 5 liters, you basically have a 100-fold improvement of the sensitivity. If you can detect 5 cancer cells with our technology in 5 liters of the human body, you have a 1,000-fold increase of sensitivity.
Give us a history of how the company was founded, as well as the financial structure.
We started out this company it's 9 months now, so we started basically in September last year, in 2012. The company was started with a number of founders and funded by a number of high net worth individuals and cancer survivors, and some of them were both. And a lot of the initial founders and investors were somehow related to the university, had somehow the relationship with the university and also with us as the founders. To this point, we raised about $1 million so far, which is enough funding for about a year or two to do the first clinical trial that we're planning to initiate by the end of June of this year. So that the plan right now is initially to test 6 to 10 patients with the technology, given that all the animal studies have been completed, all the preclinical proof of concept studies have been completed. And the next step is really to demonstrate clinical proof of concept in patients over the next 6 to 9 months, essentially. And the plan there is really to optimize the prototype and also to test a number of healthy volunteers so we can determine a baseline signal for the prototype. And then later this year, probably in Q3, Q4, 2013, we will start testing 30 to 60 melanoma patients with results to be expected in the first quarter of 2014.
Talk about your IP position.
We have 3 patents pending right now. That's basically protects the device and the method of detecting not only metastatic cancer cells in the blood, but generally speaking, objects that are floating in the blood volume, including blood clots. That's another technology we could potentially develop. But our first goal is to develop the technology for metastatic cancer cells. As we approach different milestones and potentially a strategic partnership with a larger commercial company, we could then pursue the technology for other applications like blood clot detection.
And I want to talk about some logistics facing the physicians. So, firstly, go into some detail about the ease of use.
The prototype that we have right now is a portable prototype. It's about the size of a small desk, and you can basically roll it around in the hospital. The device itself will be used in a hospital setting, an office setting. Typically, what you do is you need to have a patient either sit or lie next to the device and have the blood volume measured for about an hour. So that's something that can be fairly easily done in a hospital or office setting. And you really have a real-time readout of the results. So that's very easy for the physician or a nurse or physician assistant to handle with. So you don't really have to, as is the case right now with in vitro assays, take a blood sample, send it to the pathologist. You have to wait for a certain time until you get the results back. With our device, you have the results literally immediately and within an hour. So it's fairly easy to use.
And what case studies exist for cost and reimbursement?
The only in vitro device that's approved by the FDA for detection of metastatic cancer cells is J&J's or Johnson & Johnson's cell search system. And that's been approved for colorectal, breast, and prostate cancer. And J&J has done a pretty good job in terms of preparing the market or, basically creating the market in the oncology space. The cell search system is being reimbursed by Medicare right now, also given that a lot of the cancer patients are Medicare patients. And then also a lot of the commercial plans are reimbursing cell search. So we feel fairly confident that given that the in vitro device is being reimbursed by most plans, that if our in vivo device, which clearly has a clinical benefit compared to an in vitro device, will also be reimbursed.
And what initial markets are you targeting?
We're targeting the melanoma market right now. One of the reasons why we picked melanoma is that it's a very aggressive cancer with a high prevalence. And given the aggressiveness of the cancer, there's a lot of patients developing metastatic disease. So there's a lot of unmet need in the market for a in vivo detection system. Also, given that there's not a lot of options right now, there's not even an in vitro assay or device specifically for melanoma cells. The other reason why we picked that market is also that with our technology, we don't need any basically contrast agents to detect those melanoma cancer cells because we're using the melamine that's contained in the melanoma cancer cells as a contrast agent, which is basically an intrinsic contrast agent. I think a little bit later, once we develop that indication more, we'll probably go into other indications, other cancer types. And at this point, if we pick cancer types like breast cancer or colorectal cancer, we will probably have to use a contrast agent that we can basically develop by using antibody assays basically to use that as a contrast agent.
And so what milestones can we expect in the near term?
We're going to initiate the first patient and basically test the first patient by the end of June, and we'll have a readout of about 6 to 10 patients by the end of August of this year. That will be a fairly good clinical proof of concept trial for us. And then after that, we will start testing 30 to 60 patients in different types of stages of cancer. So the first stage would be late-stage cancer patients where we just basically demonstrate that our technology is as good as an in vitro assay. And then with the other cohort, we will test early-stage cancer patients where we'll demonstrate that we can detect a higher sensitivity than with in vitro assays. And we will see those results probably in the first quarter of 2014.
Lastly, I want to talk about some industry trends. How does non-dilutive financing help bridge the funding gap?
there's several options. I mean, there's venture debt out there right now, so we would certainly be open to consider that. And then the other option, as we enter into a strategic partnership, hopefully maybe early of 2014, if those partnerships include any milestone payments or royalty streams, we can monetize that. So that would be another option to not dilutively finance the company.
That was a company snapshot of Cyto Technologies with George Yu, President and CEO. Cyto will be presenting at the upcoming OneMed Forum taking place June 26th and 27th at the Metropolitan Club in New York City. For more information about the company, visit the OneMed Place database page.